Welcome, everyone, and thank you very much, Mark, for agreeing to give a Build a Cell seminar, and you can get started.
Sure, awesome. Thank you very much, Kate, for having me. And thank you to all of you for being here. I want to say from the get-go, I'm not fully a build a cell person. I may say things that you might disagree with or that might be inaccurate, so don't hesitate to jump in or ask questions or at any point. But otherwise, I want to tell you all about some of the work that we're doing, kind of build a cell adjacent, and also push for the potential importance of some other things that we're doing that I think, depending upon your definitions of synthetic cells, could be very important. So my group, broadly speaking, focuses on the development of biosensors, diagnostics. Typically, we envision it being for low-cost applications, but then we also very much work at the intersection of synthetic and systems biology. And so that kind of adds a flavor to the types of problems that we address and also how we address them. So, oh, hey, sorry, I forgot where I'm at. I put in some slides in the wrong order. Anyway, so big picture speaking, we use cell-free systems as the chassis for our sensors that we develop. And specifically, we largely use cell-free lysate based sensors rather than pure. And so we think that using these types of sensors and protocells, that they have a lot of exciting potential uses. So I'll tell you about how we've used protocells for multiplexed sensing that is agnostic to chemical class, meaning we wanted to be able to detect any kind of molecule. I'll also tell you about how we are planning on using protocell arrays for communication and potentially complex diagnostics. And then I also, as I already indicated, wanted to proselytize a bit to you about the importance of metabolism in cell-free systems and potentially also in synthetic cells. And so I'll start with our multiplex sensing. But before I get into too much detail about that, I want to kind of remind you about why we are so focused on sensing. So if you think about the last time that you needed to get a blood test done, right? So putting aside your at-home COVID tests, which are not really blood tests, the last time that you had to get a blood test done, you probably had to go to some big, fancy, potentially covered in glass hospital. You had to get a bunch of blood withdrawn, a venous blood draw. And then that was sent over to somebody's fancy lab with fancy equipment and well-trained personnel. And at the end of this process, you would eventually get your results, and you'd also get a big fat bill. And so, at least from our perspective, every single step in this process is kind of frustrating. And so what we're looking to do is to try and turn as many of those things on their head as possible. So what does that mean? That means, what if instead of a venous blood draw, you're able to do blood tests with just a finger stick, the same way that a diabetic might monitor their glucose levels? What if instead of costing hundreds of dollars, we could do it for pennies per individual thing that we want to assay? And the way that we want to do this, and what if instead of needing to be in a centralized facility with a lot of expert personnel and expensive equipment, what if we were able to do it in arbitrarily remote places, some of which might not have the same resources that our advanced clinical hospitals have, and some of which might not even have reliable access to electricity? And so the way that we look to do this is by harnessing bacteria, by harnessing life, and trying to turn the existing parts that are out there and new engineered parts into the potential for new sensors. Now, we used to be on the whole cell side of developing biosensors, and we've since shifted a lot of our efforts into cell-free space. And so when we talk about cell-free protein synthesis, let's see if I can find, when we talk about cell-free protein synthesis, what we mean is that we're going to take our bacteria, we're going to bust them open, we're going to get rid of the genome, we're going to get rid of the cell membrane, and what's left is this highly capable reaction mixture that can perform a lot of the functions that are otherwise possible in cells, including, importantly, transcription and translation. And what we have done is focused on making customized cell-free sensors that can detect biomarkers of interest. So what we're doing is we're turning these cell-free lysates into essentially the equivalent of a mass spec, right? And so how can we make these cell-free lysates be as sensitive and specific as possible for the things that we want to measure? And when we measure them, we can have fluorescent reporters, and that's kind of the go-to for a lot of engineering biology and synthetic biology. But we also want to think about how we can do things that might be more field friendly. Because again, remember I asked at the beginning, can we do this in places where people don't have access to expensive equipment and might not even have reliable access to electricity? Well, doing something that's a colorimetric reporter would enable you to do that, right? Because then in that context, it's not really that much different from a pH strip, which, you know, everybody, not everybody, a lot of people have done in high school chemistry class or even in arbitrary parts of the real world. Like if you ever had to like check and see if your pool was safe. So that's our kind of big picture goal, is to use these cell-free systems to make these customizable analyte-specific sensors that give colorimetric outputs that can then be deployed to arbitrary places in the world. And we're definitely not the first people to do this. There's a bunch of people that have done work by adding DNA and RNA programming so they can measure and detect the presence of pathogens, and specifically pathogen DNA or RNA in a sample, and they can get a colorimetric output of purple it's there, yellow it's not there. And so there's been some great work in this space. And where we're focusing on our niche is that we're not typically looking for presence and absence detection when in the problems that we're looking at. Whereas a lot of the nucleic acid sensing pathogen detection is about, is the pathogen there or is it not there? But for things like nutritional biomarkers, for a lot of other biomarkers, it's not the mere presence or absence of something, but rather it is the semi-quantitative level of it. And so that's something that we try to focus on. And then also we try to focus on equipment-free quantification, right? Because it's maybe easier to equipment-free see presence or absence, but getting that quantification equipment-free context is quite challenging. And as I've already mentioned, a lot of the scope has been focused on infectious disease and often uses kind of heavily processed bodily fluid samples where you wind up having a very small fraction of your actual sample that's added is the original sample that was taken from the patient. That implies a lot of extra steps that would add complications to doing this in the real world at the point of care, at the point of need. And so when we think about the things that we might want to sense when we have these types of blood tests, whether they're being done in the lab or whether they're being done out in the field at the point of need, we realize that there's a lot of different types of molecules that we would want to test. And when you think about how people currently measure these molecules, let's say in a clinical lab, well, nucleic acids, they're going to measure with something like qPCR. With a small molecule, they might use something more like a mass spec. With a protein, they might use an ELISA or something else. And so you see very different pieces of equipment that are required to measure these very different types of molecules. And so one of the problems that we thought about trying to address in this context of cell-free based sensing is, could we maybe measure all of these molecules even in the same sample with the same readout? And so that was one of the driving questions in our work here that was led by Yen Zhang, who goes to a bunch of build to cell stuff as well. And so basically she wondered, can we take our cell-free systems and can we add some kind of phase separation element that will allow us to perform highly multiplexed analysis of different biomarkers? And she did a great job executing this. And so I'm going to talk about some of her work here. So she started off with the idea that polymer two-phase systems can enable compartmentalization of cell-free systems and thus cell-free sensors. So when I'm talking about polymer-based two-phase systems, you know, you can start with oil and water, or oil and vinegar, right? If you've ever had salad dressing, you know, the phase separate out. If you shake them up, they'll kind of mix up, but they're still phase separated, and eventually they're going to separate out, right? So organic and aqueous, those I think we're more used to seeing in the real world. These things phase separate, two distinct phases. But you can actually do the same thing with two aqueous phases if you add two different types of polymers. So here we have dextran and PEG that they do not want to be together. And so they will spontaneously demix and phase separate into two different liquid phases that are actually both aqueous phases. And when you think about things in the context of what is a friendly and permissive environment for a cell-free system for transcription and translation to occur, it probably is going to be in an aqueous system. So this kind of gives us our first step of how we can enable compartmentalization of different cell-free reactions within an aqueous two-phase system. And so the thought process would be that, you know, at the beginning, I would add my sample, and it would maybe be distributed throughout initially, but there would then be perhaps some kind of partitioning that might happen if you have nucleic acids and if you have proteins. But maybe sometimes you won't necessarily have partitioning. So you might have different levels of sensitivity that you get. But in this context, we hope that we'll be able to create different sensors that might detect our proteins or might detect our nucleic acids or might detect our small molecules that will enable us to get our measurements. And I want to jump in since we do have questions here. When we have a PEG-DEX system, does the entire lysate compartmentalize to one phase, or is it possible that some components partition to one phase and others to another? So it turns out, and actually we'll show as we go along, that everything sufficiently compartmentalizes that is critical. And actually, you know what? I think we don't go into all that detail. So let me put a pause on that to see how far into the detail we get, and I'll make sure that I follow up with a full answer there. So what we do is we do this compartmentalization such that we imagine that, okay, I can get one droplet that has one sensor in it, another droplet that has another sensor in it. And if I could just physically separate these droplets, that would enable me to have a multiplex system in the same sample. So if you look at it maybe from the side here where we can see sensor A and sensor C, you would load in your target overall and here into your PEG phase and your bulk phase. And your cell-free reagents would be essentially compartmentalized and constrained within these DEX droplets or phycol droplets. And then you add your sample and your sensors respond to the analyte either diffusing into or being partitioned into each individual sensor. And so only the sensors are going to turn on that are capable of sensing the analytes that are there. And so we showed that we could do this, and actually this is probably the best place to point this out because I don't think I have it later on. All of this cell-free stuff, all the components that are necessary for, let's say, all the macromolecular components that are necessary for transcription and translation, if we start them in the DEX or phycol phase, they will stay there. I think it's possible that if we waited long enough and then went to try and measure them elsewhere, we might find some, you know, equilibrium partitioning that some fraction of it leaves. But over the time scales that we care about in our sensors, they all stay there. However, small molecules do not tend to compartmentalize as strongly. And so what we found is that we can't just add completely just sample here. We need to also include essentially the energy mix because those small molecules are not going to preferentially go into our droplets or even stay in our droplets. If we start off with the small molecule energy mix in our droplets, we find that they are going to fairly quickly equilibrate. And given that these droplets are like 1 microliter in the span of 100 or 500 microliters, you can imagine that the tremendous dilution in small molecule concentrations that we get becomes problematic. And so we actually do know that we need to supplement with additional small molecules such that when we add the final complete sample volume, we'll have an appropriate concentration of relevant small molecules in our compartmentalized sensors. And so we showed that we could do this, and we demonstrated multiplex detection of multiple clinically relevant analytes. A lot of our biosensor work to date has focused on nutritional deficiencies, specifically micronutrients like vitamins and minerals. So we took two previously published sensors, one for zinc and one for vitamin B12, and show that they can work in these droplets right alongside of pathogenic nucleic acid biomarker detection, where in one of them we detected DNA, and another we detected RNA. And so we showed that these all can actually work together. And I don't have all the details of every single step along the way. It's in Yen's paper in Nature Communications that talks about this work. But kind of the big picture about the success is that when we have none of the reagents, or sorry, none of the targets that are in a reaction, we get kind of some low baseline. This is imaging of the results using a ChemiDoc, not a plate reader. And then we also did some image processing to get some quantification out of it. And so when we have nothing there, there's a little bit of background, of course. And then when we add, say, our zinc and our DNA for one type of pathogenic DNA detection, we see we get very strong signal out of both of those, and not too much difference in the background there. Frankly, most of the increase here is coming from bleed of fluorescence, that there's some small degree of leak out of the GFP that's happening. And then similarly, when we add our vitamin B12 and the RNA for our other pathogenic target, we see that we can get signal there, which again can be quantified. Here you see that the amount of signal that you get from any one sensor, the same way that if you took this sensor and you implemented it not in a protocell format and not in an array of protocells, that you might get different signal to noise ratios. And we see that here, right? This STX1 switch, we used a toehold switch based sensing approach here. That one is just not as strong. We chose one that had lower leak and maybe less activation, but still also demonstrated that even with a lower degree of expression, that our approach can work. So this process does work, and it's pretty cool and exciting for us. And we can also detect them all when they're there. But I do want to highlight here, kind of at the beginning, not at the beginning, but at the beginning of this section, where I said, hey, nucleic acids, small molecules, proteins, if you want to detect them, you're going to need three fundamentally different pieces of equipment. But now, in the same sample, we're able to detect those three fundamentally different types of molecules with the same reporter, with the same simple piece of equipment. So we've kind of obviated the need for a lot of other complex equipment, which, as you can imagine, makes this more point of care or point of need friendly. And then to move even closer to that, we also showed that we can do this in a freeze-dried format and that we can get a visually interpretable readout. So here what we did was we stuck to just the nucleic acid detection, just for the proof of principle here, but we could easily do this with small molecules and proteins. So we stuck with just our nucleic acids here, and we had three different nucleic acid targets, and we freeze-dried them down. We lyophilized them down, and we included an XGAL reporter, which is well known for blue-white complementation assay. And we essentially use that to get a colorimetric readout using the LAC-Z gene, which would then be expressed in response to the presence of these different nucleic acid targets. So we freeze dried them, and then when we add our sample and then wait a little while, we can see that when we add the individual types of nucleic acid sequences, that we get the respective visually interpretable signals, right? So at least from a nucleic acid context, it's very clear, either present or not present. The blue-white is definitely more of an on-off approach. We have other approaches that we use in our lab for more quantitative readout, which you can imagine would be much more important for things like zinc and vitamin B12. And we did some investigation as to how feasible those are. There are some diffusion limitations. By that, I mean the opposite of diffusion limitations, which is that some of those reporters that we use can actually diffuse out of our cell-free droplets, whereas this XGAL, once it gets transformed, it actually precipitates out and then stays in the droplet, and so that made it a little bit easier for our purposes. But moving forward, we'll look for better ways to get quantification into these approaches. Speculate on boundary conditions for multiplex detection. How chemically different do the analytes need to be for reliable multiplex detection? So the question is, how different do my analytes need to be in order to get reliable multiplex detection? And I hope it doesn't seem like I'm bailing out on the question. I think ultimately that answer depends on the sensors that are being used. There is nothing inherent to the physical compartmentalization into different drops that requires those target analytes to be any more similar or more different. It ultimately comes down to what sensor are you implementing in those droplets. And if your sensor has a lower degree of specificity, then that is going to constrain and require you to have more different analytes, sorry, more chemically different analytes than you otherwise might need if you had a more specific sensor. So I don't think that the multiplexing puts any additional constraints onto how similar or different our analytes need to be. In terms of detection level interaction, I think the best place where you can see that is, if I can jump back here, that there is arguably some essentially diffusion-based, reporter diffusion-based detection level interaction. And I think that's where we're going to see most of this additional signal, off-target signal that happens. You could also imagine so these were done with plates that our collaborator Shu Takayama, which I should have highlighted his involvement earlier. These are plates that he already had made for kind of other purposes. You can imagine redesigning these plates such that the distance between these two different wells is a little bit different, and such that if it's twice as different and your diffusion time is going to scale with the square of the distance, then if you just make them twice as distant, then you'll give yourself 4x more time before you start to see any of that diffusion. I think it's mostly diffusion-based stuff there. Why is the signal maxing out at the same levels for all? I think that's ultimately a limitation on the Chemi doc. Yeah, I think that's basically, yeah, that would be my interpretation there. I'll say that for those, you don't necessarily see the same maxing out of signal. And as I highlighted earlier, that one definitely is not the same signal level. Yeah, so I think otherwise that's an instrument limitation. Cool. So that's our multiplexing stuff. And there's more details to it. I'm happy to answer more questions about those details there. I want to talk about some ongoing work that we're not as far along in yet, but I think is kind of synthetic cell relevant. The idea of using protocell arrays for communication and complex diagnostics. So this is work that's being done by Felicia, who's a current graduate student in our group. And she's basically asking the question of, okay, what if I want to take this multiplexing and what if I want to apply it to an actual diagnostic task? And so specifically, maybe let's look at trying to diagnose pancreatic cancer. And maybe let's say that I know that these are four proteins that have levels that have these are four proteins that are known to be biomarkers for pancreatic cancer. But it's only a biomarker if these three go up and this one goes down. And that's then a good panel that gives you an indication of some degree of specificity for pancreatic cancer. So if you can imagine taking our current approach and saying, okay, what if I'm able to just set a threshold and say if my CRP is above some level, it'll turn on. If my albumin is below some level, it'll stay off. And so if you imagine then what your output could look like, it could look quite confusing, right? So each of these sets of four is one potential set of outputs. And only one of these is going to, which one is it? This one. Only this one is specifically going to tell you that this is the pathologic condition that you're looking for. Otherwise, this might look scary, but it's not actually the pathologic condition that you're looking for. So you can imagine, say, okay, well, I'll just give somebody a key that says, okay, it should be this shape when you come out, when you get all of your signals, and this shape is good, and this shape is bad in terms of like which different dots have lit up. As we start to think towards, can I make this more field friendly? Part of being field friendly is being user friendly. And if you can remember the last time you actually did do a COVID test, the excruciating detail and efforts at oversimplification that go into the design of these tests to make them robust to different users. Telling them you should be looking for this shape, but not this shape is probably asking for trouble. And so we wondered whether we can somehow make this simpler. could we borrow parts from how bacteria communicate and allow essentially each of these protocell droplets? And I'm sorry, very early on, I maybe referred to these as protocells. And this was a great point that Shu made as we were working on this, because I just looked at it as we were compartmentalizing biosensors. But what he pointed out is that these are in some way membraneless protocells, right? That they're separate sets of transcription, translation that are separately programmed, that can execute functions. And in some ways, maybe not a complete synthetic cell, but a kind of proto-early synthetic cell. And so since we're thinking about protocells, maybe we can borrow from how bacteria communicate. So, bacteria might want to say hi to each other, and they can say, okay, well, this person might have levels of elevated levels of CRP. And so how can we send that information from one bacteria, one protocell, over to another protocell? And so the way that bacteria do this is they might create, let's say this is a great quorum sensing type example. They synthesize some quorum sensing molecule inside one cell, and they can communicate to other cells. They might be a different type of cell, or it might be other cells of the same species. This molecule diffuses out over to other cells and then diffuses into those cells. And then inside those cells, they have a transcriptional response to the levels of that molecule that can then start the expression of some reporter. And so we're trying to borrow that. We're trying to say, hey, can we use, whether it's second messengers within a cell or whether it's known extracellular, intercell communications, can we take these and harness them for communication within our protocell array? And so the thought process is that we can take this approach and create a kind of multi-input, single output system where protocell 1, protocell 2, protocell 3, they each create some small molecule. And then on the back end, you have this integrator protocell where the small molecules all diffuse to your integrator protocell. And that integrator protocell has the transcriptional response for all those different small molecules, such that when that small molecule is there, it expresses some reporter. And then critical to this is using some previously published cool stuff of a highly fragmented T7 RNA polymerase, where you can break it into, we're just using 3 pieces. I believe the publication that we're looking at had over a dozen pieces, where any one of these 3, or actually any combination of these 3 alone, will not give you expression. It will not give you a functional RNA polymerase. But when you express all 3 of these fragments of a T7 RNA polymerase, they reassemble and allow for expression of reporter. And so that's the approach that we're taking. And I thought I had another slide there. And so our thought process then is, we have all these sensors that we previously developed, right? We've developed a vitamin B12 sensor. And what we've done previously, we said, okay, well, when we detect our vitamin B12, we'll create some GFP output. Or people have developed nucleic acid sensing modules where they produce GFP. Or we've separately, and I'm not talking about this today, recently reported a new approach for protein sensing in cell-free systems, where we get a colorimetric readout. So instead of doing these different readouts, let's instead change these readouts, these reporters, so that they're not immediately visually interpretable, and instead so that they create the small molecules that can then go to an integrator protocell that can then interpret those and create a visually interpretable output, right? So that then those, so instead of producing GFP in response to any of these sensors, we produce the genes that will allow for production of the small molecules in our bacterial communication cascades. And so that's instead what this looks like. We can imagine putting protein detection in front of the enzymes for producing a homoserolactone. We can imagine putting nucleic acid detection in front of the enzymes that are necessary for creation of cyclic AMP or cyclic di-GMP. And so then that allows for small molecules to be produced that then migrate over to our integrator protocells. So a lot of the work, I mentioned this is kind of work in progress. I want to give you just ideas of how we're thinking about protocells and synthetic cells. A lot of stuff we've been working on up until now has just been doing the cloning, validating the transcription factors, validating the metabolic enzymes. And so we showed that, yeah, our transcription factors, they do in fact work when we get them into protocells or protocell arrays. We actually find that sometimes they work better in a protocell, and sometimes they work worse in a protocell. And we're still trying to identify the landscape of when we should expect those two different possibilities to happen. We also showed that we can get good response in our quorum sensing transcription factors as well, using a homoserine lactone. We also recapitulated the previously published fragmented T7 RNA polymerase and showed that you can have any combination of a pair of them and not really get much expression, but when you have all three of them, we have a tremendous signal. And so we've shown that these pieces can work in our hands, and we're currently working on putting them together. And so we're not as far as that as I otherwise might have liked to tell you about today, but it's exciting stuff that I think is pretty cool moving forward. And I look forward to us being able to put those pieces together in order to make potentially complex diagnostics that can still be easy for the user, yet still attack different and difficult diagnostic problems, since frankly, in a lot of biomedical diagnostics and in clinical diagnostics, it's not often just one single molecule's presence or absence or levels that defines a pathological condition. It's typically multiple. So I'm going to answer Lucy's question with respect to our previous protocell work. We did actually get to do that in complex samples. We did it with plasma, not whole blood. We're still working on how best to do this with whole blood, but with plasma, we did show that this works. And in the 3-part T7 work, this is all in E. coli lysate. And we're expressing the parts with sigma 70 or some other endogenous promoter. I'm trying to think about what parts there. So we can express that's not quite accurate there. We use both T7 and sigma 70 promoters to express individual parts. I kind of glossed over the fact that this fragmented polymerase uses a different orthogonal promoter system. So we're still able to use regular T7 plus this fragmented polymerase there. And I think I could maybe talk extra or later about prep process for lysates. I'm going to put a pin in that. We can come back later, because I do want to talk about one other thing before I force myself to run out of time here, because I mentioned along the way that we're using these metabolic enzymes to create the small molecules. So that gets me to thinking about metabolism in cell-free systems. And I want to kind of highlight for people that metabolism very much exists in cell-free systems, right? Metabolism exists in cells, right? And let me remind you, when I say metabolism, I mean the process by which small molecules, nutrients, are turned into energy and typically the building blocks for more cells. And so metabolism is definitely there in cells, but it's also there in cell-free, right? So when we're doing these lysate-based systems, and so if you're a pure person, then this is maybe less applicable to you, but if you're doing a lysate-based cell-free system, you are taking a snapshot of all the macromolecules in the cell at the time when you harvest them. That includes the entire proteome, which includes the entire metabolic proteome. So all of those metabolic enzymes, they are there. They are waiting to do stuff, whether you think about it or not. And so that's what I want to talk about a little bit right now, is that metabolism in lysate-based cell-free systems is absolutely there, and it is absolutely important to how those cell-free lysate-based systems work. So our first discovery was done by April, who's one of the alums from our group. We saw something interesting about cell-free protein synthesis. I mentioned earlier, we kind of exist at the interface of systems and synthetic biology. We were getting into the cell-free space about 10 years ago, and we were thinking to ourselves, like, I also do metabolomics. I wonder what happens when I metabolomics these samples, right? And so we decided to do that, and we saw some interesting things that happen. Oop, there we go. So one of the things that we found was that if we took our cell-free lysate and we either did expression or didn't do expression, so here what we have is our colors represent time points and our shapes represent, is there a DNA template or is there not a DNA template? And what we see is that a major separation based not on is there DNA or is there protein being expressed, but rather what time is the measurement being taken at. Now, I do want to take a second to pause because I've put this up here. This is a PCA plot. This is essentially the lingua franca in metabolomics. And I want to briefly explain to you how to interpret a PCA plot if you haven't seen this before. So a PCA plot is a way to take high dimensional data. We measure tens to hundreds of metabolites, and we want to project that into only the two most important dimensions that are each linear combinations of a bunch of different metabolites. And PCA is a mathematical transformation that allows us to find kind of like the eigenvectors. It allows us to find the combinations of metabolites that best capture the variation across all of our samples. So we'll get one principal component, which is a linear combination of all our metabolite measurements for any individual sample. And then we have another one that is not quite orthogonal, but fundamentally different from the first principal component. And these can capture 34%, 12% of the variation across all of our data, right? And so the way to read a PCA plot is basically when you know what samples are what, when you see circles that are overlapping, it means those are extremely metabolically similar. They have highly similar metabolic profiles. Because what we're doing is we're using GCMS to measure small molecule levels in all of our different samples. So when you see these two overlapping, it means that the metabolite profile of these is essentially identical. When you see these orange and green are not overlapping, it means there are fundamental differences in the metabolite profile between these two situations. And so now zooming back into what we're looking at here, the places where we don't see a difference is whether we have DNA or not DNA, which means whether there's protein expression or not protein expression. There is a trivial metabolic difference. The real metabolic difference is whether you're looking at the beginning of your reaction or at the end of your reaction. And I have this as lysate NN. This is one type of lysate processing strategy that we used. We looked at 3 other different lysate processing strategies and always saw the same result. And then this is just looking at 2 time points. If we dug in and looked at one specific lysate at all these different times, you see that if you're able to track red, purple, blue, green, yellow, you get this kind of pathway in time where we're moving throughout these 2 principal components in a fairly continuous fashion. But at every single time, the 2 shapes within the color are always identical. So time has the biggest impact on the metabolite profiles in our cell-free systems, not protein synthesis, even if we have very high levels of protein expression, which to me was a little surprising initially. And so we wondered, we're seeing metabolic changes. Do these metabolic changes actually have an impact on protein synthesis? And this data suggests that it does. Let's dig in a little bit. If we take just a cell-free reaction and we add the plasmid 8 hours after we put our reaction together versus only 4 hours after we put, versus immediately after when we put our reaction together, you see that your expression levels drop precipitously. And this even happens if you spend these 8 hours cold, right? And even more worth noting, this is not because our equipment is falling apart. Why do I say that? It's not that our polymerases and ribosomes are freaking out, because if we do dialysis here, look at these blue and green. I meant to change this to also include the blue and green curves here. Dialysis is essentially, which is part of the workup for some of the lysate prep protocols. Dialysis is essentially a really long incubation step. And so we go through this long incubation step, and even with dialysis, we still see very high expression when we add the plasmid immediately upon reaction assembly. So it's not inherent instability of the expression machinery, it's something else, right? And for us, this suggests that endogenous metabolism, and that's what we're referring to this as, the fact that proteins that we're taking over with the lysate, they're ready to do metabolism. And this endogenous metabolism, it appears, is actually hurting protein synthesis if you just let it go on at the beginning. And so Soar, who's a current PhD student in our group, he's picked this up and he's running with it in like 15 different directions and doing some super cool stuff. And one of the things that he found is that endogenous metabolism actually seems to vary significantly depending upon the lysate prep methods. So we have a bunch of different lysate prep methods that we refer to as Jewett and Nero protocols, because those are the folks that originally published them. There's some big fundamental differences. And even if you ignore the dialyzed versus not dialyzed aspect, the Nero and Jewett protocols for the analyses that we're about to do gave very similar levels of expression, but their metabolism wound up being very different. And why do I say that? What we're looking at over here, these are all your 0 time points. And then these are all your, I believe, 4-hour time points. And so when you look at the Jewett preps, you see they behave in a very similar way. They just move along principal component 1 from time 0 to time 4 hours. There's some degree of variability, but it's not substantial. Whereas the Neuro prep started in the same place and yet wound up in a very different place than principal component 2. So we see that the endogenous metabolism is somehow inherently different. The metabolic processes are happening differently in the Neuro and Jewett preps, probably due to different stability of different metabolic enzymes. We haven't dug in that deeply yet, and we're working on that. And I do highlight down here, everything from here on out is completely unpublished, and so some of this is maybe a little rough around the edges at this point, but it's cool, exciting, not quite off the presses, but more recent stuff that we're really interested in. So lysate prep methods affect endogenous metabolism, which we've already shown can affect total expression. Even the reaction format affects endogenous metabolism. And by this, I mean, when April did her work, she did it all in Eppendorf tubes. So she would do a cell-free reaction because, we're used to doing cell-free reactions in 10 microliters, but we're not going to be able to do mass spec on 10 microliters. We need 10x more than that, if not more. And so we did it in bigger Eppendorf tubes and not little PCR tubes or microwell plates. And so when she did this, she found what we saw before. As time goes on in your metabolic profile, whether you're with or without expression, you don't see any difference in metabolic profiles. Soar, instead of doing it in Eppendorf tubes, he wanted to simplify things for himself. He wanted to make it easier. He did it in 96 well plates. And he saw, what I thought was initially kind of crazy, because when he didn't have protein expression, this was the metabolic pathway. And when he did have metabolic expression, he got a totally different pathway of metabolism through this principal component space, which is at odds with what April previously saw. And at first, when he showed me this, I was like, dude, you're doing something wrong, right? And he did it again and did it again, and kept seeing it until only upon deeper discussion did we eventually realize he was doing this in well plates and April had been doing it in Eppendorf tubes. He goes back and does it in tubes and reproduces exactly what April saw. So the format seems to have an impact on metabolism, which we wondered was maybe about oxygenation, right? You had a permeable film on top versus sealed tubes. And it turns out if you try to measure via GFP protein expression in different oxygenation conditions, whether tubes versus plates, whether agitated versus non-agitated samples, that in all cases where you expect to get better oxygen transfer, we get better expression, right? So these red cases are the ones where we expect to get better oxygen transfer. So our cell-free expression is being affected substantially by oxygen limitations coming from metabolism. And I'll note that this doesn't have to do, we have separate experiments that I'm not showing you the results of here. This doesn't have to do with GFP maturation needing oxygen. We've shown separately that it's not due to that. And so similarly, when we look at agitation, that also affects the metabolome. We see a fundamentally different metabolic journey through principal component space if we have agitated cell-free expression versus not agitated cell-free expression. And then what I think is maybe one of the most interesting recent discoveries is that looking at all this, Soar then said, okay, so metabolism is apparently huge. He looked and found some things that were running out towards the end of our reactions and wondered, okay, if I'm running out of some of those things, maybe if I supplement with some of them, I'll get better cell-free expression. And amino acids, we were running out of some amino acids. So he said, okay, let me just take the whole amino acid mixture and I'm going to supplement with some amino acids. But the weird thing that he saw is that when he let the reaction go almost to the end and then supplemented, that helped. But when he supplemented at the beginning with essentially 2x amino acids, that hurt. He got less total expression. And so he went through and said, okay, let's figure out all the different times when I can supplement with 2x amino acids. And if he does it before this point in time, he gets less expression. If he does it after this point in time, he gets more expression. And then super cool to me and very exciting for Soar, because this is kind of the thing that he came in most interested in. This time where it transitions, that's actually when you look at a cell-free GFP expression plot as a function of time, that's where it hits the inflection point and expression starts slowing down, which for us is a clear signal like, I wonder if something metabolic is going on there. And then kind of building on that, he also showed that, okay, if I just add if I add amino acids at the end, if I take half of my amino acids and add them at the end, and starting from 1x amino acids, sorry, I'm not articulating this well, and I apologize. He found that you can use the same amount of amino acid, you can get the same level of expression by from only 70% of the amino acids by doing some at the beginning and then some at the end. And so that to us is an indication that, okay, there really is a metabolic impact here because I'm able to actually decrease the total amount of amino acids that I'm using and still get the same total expression only by adding them at different times. So for us, it's just that there's lots of crazy metabolic stuff that's going on. And as you start to think forward in making synthetic cells, if you're using lysates ever instead of pure, you should perhaps be thinking about how metabolism might affect the things that you're trying to do. So that's the story that I wanted to tell. I'm sorry I ran maybe a little over what Kate had initially suggested as my high end, but it did also answer some questions in the middle too. So maybe it all comes out in the wash. But I told you about how we can use protocells for multiplexed sensing across diverse chemical classes. And then also that we can use these protocell arrays potentially in the future for protocell communication and complex diagnostics. And then finally that metabolism I encourage you to think of as very important. And so the last thing I want to do is thank these folks that actually do all the work. Bolded here, and I tried to highlight them as I went along, the people who did the work specifically that I presented here. And it's all in an effort for us to create more quantitative and in-field cell-free sensing with minimal equipment. So I will end there. And I'll maybe start asking or answering the remaining questions and then also be open to new questions as well. So Charles asked, how intensive is the prep process for lysates? The answer is intensive. It takes a bunch of effort, and being trained on it well the first time is important. And you need to get good at it to do it reproducibly. But it is something that you can train your average undergraduate to do reproducibly. Would it be correct in assuming that a separate culture would be needed for each sensor lysate to be used in the array? So that's actually not true. Or it can be true if you want it to be. We can use the same lysate for a bunch of different sensors where keep in mind that the lysate is added to all the sensors, but the DNA programming for the transcription factors, et cetera, is different in each of the different droplets, right? So we don't need to prepare a different lysate. We do need to prepare a different mix of lysate and DNA. However, if you really wanted to, and this is one of the strengths here, let's say it turns out that one type of lysate works best for a zinc sensor, a totally different processing of lysate works best for B12 sensor or nucleic acid sensing. We can actually put those different types of lysates into the same multiplexed device, and that can all work, which I think is pretty cool. Shelf life, I'm going to punt on that question and say that there are people out there like Brad Bundy who do a great job of identifying what the shelf life is for these things once they're freeze dried and improving that shelf life via lyoprotectants. It's something that we focus less on in our upstream development and that we'll come back to as we get more to translational implementation. What I will say is that at room temperature for these currently, when we don't use any lyoprotectant, you're going to get less than a day before you're down to like 10% of function. But Brad and other folks have shown that with appropriate lyoprotectants, you can keep 80% function with like a week or so. And this is different than pure-based approaches. Those can stay for an extremely long time. Henry says the tube versus plate result is wild. Agreed. How do I think that affects reactions in the droplets or liposomes? Awesome. Yes, that is exactly where we're at, right? Of like, okay, so is the depth to which we have sample on top going to affect our expression. And for our protein sensors that I didn't present, some of those are very sensitive to kind of like the minor aspects of the reaction assembly, and whether that oxygenation could affect how good of results we get from that. It's a fascinating and important point that we need to keep in mind as we develop this platform further. Kate asked a random practical question. How do we do 2x amino acids? We can barely 2x with that. The 2x amino acids is coming from the same stock, but just adding twice as much volume in the initial cell-free reaction and removing some of the other volume that would be allowed for water. Oh, sorry, that was your second part of the question. That's exactly what we do, Kate. Kirsten asked, is all this work done in E. coli lysate? Yes. Have we tried other types, eukaryotic or other bacterial? We haven't, and I think it would be super cool. It's just not a space that our lab plays in a lot right now. We have one student who had been working with an undergraduate to try and create a yeast lysate based off of some Jewett protocols that was working for us for a little while. But then we ran into some difficulties, and that student hasn't yet picked up that side project again. So we do not have, oh no, I'm lying. Lex has some stuff buried that I don't get to see the data much about, where she uses a couple of other different lysates. We've never done any of them in protocells though, and definitely never in protocell arrays. But we absolutely could do that. We just haven't gotten there yet because it's not a prominent thing in our lab. Other questions? Feel free to type or unmute and ask.
I think Taesok has a question.
Oh yeah, go for it.
Mark, great talk. I actually know, I mean, your earlier part of your presentation, including Jan's works, was still exciting, but the last part is actually the most exciting to me, because that potentially lead to multiple different applications in the end. But one thing I want to ask is, probably I missed that part. So you didn't see any difference between DNA in without DNA, right?
When we do the expression in a closed Eppendorf tube, we see no difference. When we do the expression in a 96 well plate with the same volume, but with a gas permeable seal, we do see a difference, and we see different metabolism with and without protein expression. And so that was like crazy to us. I mean, sorry. And initially it was crazy to us. Then we realized like, okay, it's ultimately an oxygenation issue. And so SOAR has work that I didn't cover here, where he's tried to figure out like, is it a diffusion length issue? And he's kind of like tried to figure out, you know, is it total concentration versus diffusion length? And we're still working our way through that in more detail.
So without DNA, you see the difference, right?
Correct. Yeah. When we do it in a format where oxygen is in greater presence.
Okay, so that makes sense. I mean, of course, I mean, you know, different, you know, oxygen conditions should have different, you know, expression level. But my question, real question is, based on this crazy data set, what insight you giving us? And then I also see, you know, you didn't see any difference between DNA with DNA without DNA. That also potentially means you could harness a lot of potential sensors in this kind of, you know, metabolite, you know, enzyme pools. I'm not sure, you know, what is your insight out of this kind of, you know, crazy data set? What do you want to do next? I mean, that's what
Yeah. Yeah, sure. And so SOAR's big picture vision is maximizing biomanufacturing potential. What I would say is, in short, this suggests that we can do metabolic engineering on cell-free systems in order to improve them substantially. Because what I envision is, so when we look at the cell-free system, you do protein expression. You definitely don't get as much protein out as you got amino acid in, right? But we do find that we run out of specific amino acids, and it's not because all those amino acids are being incorporated. Some of them are just being metabolized by the rest of metabolism. So you could imagine going in and engineering your chassis cell and doing classical metabolic engineering of clipping out specific enzymes or overexpressing certain enzymes that's going to reshape metabolism and enable you to get faster or better cell-free expression, which could just translate to better protein expression, or it could potentially also translate into biomanufacturing for small molecules as well.
I see. But you got this kind of the PCA plot by measuring metabolite, right?
That's right.
So based on that, also based on the difference, what I thought, because also at this time, I thinking, you probably mentioned briefly without DNA, there is no difference. My kind of guess was there are some, some enzyme that potentially regulate by the metabolite as well. In that way, you could also harness those kind of sensors, potentially a lot of sensor actually, that is regulate by the metabolite. That's another great asset later on for a lot of community members. So that's why I'm asking also that question as well. Metabolism clearly different, so I have clear idea what you want it to do, but I also thinking about you could also harness some potential sensor out of this kind of exercise.
Yeah. I see where you're coming from. Definitely. I think consistent with both who you are and who I am. You look at this as a positive opportunity, and I look at this as something I got to worry about, right? And so, like, I look at this and I say, like, oh my gosh, there's going to be some crosstalk or interface between metabolism and the thing that I'm trying to measure. But I agree wholeheartedly that you can actually harness this and turn this into a benefit for the total system, whether it's specifically a sensor, right? Like Lucy just said, like a metabolomic signature as a readout, or more broadly. To get back to, me and I worry about things. So this metabolism issue actually is a problem in our bacterial communicate or protocell communication work, because one of the things, I can't remember if it was cyclic AMP or cyclic di-GMP, one of those is actually getting metabolized in the cell-free lysate as we synthesize it using the enzymes that we're adding in. And so this is another, like, okay, metabolism is there, and you need to be aware of that and somehow tweak what you're doing. Otherwise, we're not going to be able to communicate with that molecule between our different protocells. So yeah, but agreed. Like, if I look at it from, I always admire your positive outlook on things. And so, yeah, I agree that we can turn this challenge into a benefit for our science overall.
That makes sense now. Thank you.
I think these are all the questions.
Cool.
And I just want to say thank you so much. That was really inspiring, especially as someone who deals with extract problems ourselves.
Cool.
I
Yeah,
really
thank you
love
very
it.
much for having this. This was fun. I hope you don't mind me like stopping in the middle to answer questions, but
Oh, I think we had very briefly, I saw someone had a hand up and
sorry, amazing talk. I just misclicked it. I wanted to clap.
oh, okay.
Yeah.
Awesome.
All right.
Well, thank you all for having me. I appreciate it.
Thank you, and have a great rest of your Monday.
Thank you. You too.
Bye.